Welcome, everyone. I am your teacher for today, and we are jumping right into a really fascinating topic in biology. Today, we are going to explore the absolute wonders of how living organisms create new life. Let's get started, shall we?
Created by MindStick's Foundation Trust.
Here's a quick look at what we'll cover today. First, why do organisms reproduce at all? Then, we'll go through the basics of asexual reproduction. From there, we'll see how it happens in unicellular organisms and then move on to multicellular ones. And finally, we will wrap up with vegetative propagation and spores.
Okay, let's dive into this first crucial topic.
So think about this for a second. What helps maintain the continuity of a species? We have already studied essential life processes, right? Things like nutrition, respiration, excretion. Now, those are absolutely critical for an individual to stay alive. But reproduction? It's completely different. It doesn't actually keep the individual organism alive. Instead, it is the magical process that ensures the species itself doesn't just disappear from the Earth. It's literally what keeps life going generation after generation.
Let's move on to the core biological concept of our lesson today, the basics of asexual reproduction.
Please make a careful note of this definition. Asexual reproduction is the formation of a new organism by an organism of the same species, completely without the involvement of gametes. No male and female reproductive cells joining together here, none of that. It is strictly uniparental, meaning only one parent is involved. And because it's just the one parent, the whole process happens purely through mitotic cell division.
Now, naturally, every biological process has its pros and cons. The biggest drawback here? A total absence of genetic recombination. The new organism is practically an exact genetic copy of the reproducing parent. There is zero mixing of traits. But look at the advantage. It is incredibly fast. When conditions are favorable, organisms can multiply at a staggering rate. To put that into perspective, certain bacteria can double their entire population in just 20 minutes. It's super quick.
Right. Now that we have the basics down, let's scale things down to the microscopic level and look at single-celled organisms.
In unicellular organisms, the most common method is binary fission. The parent cell simply divides to form two similar daughter cells. But here's the catch. The axis of this division actually changes based on the organism's shape. Take the amoeba. It doesn't really have a specific shape, does it? So it can divide in literally any plane. We call this simple binary fission. Paramecium, on the other hand, divides horizontally, which is transverse binary fission, and the euglena divides vertically, making it longitudinal binary fission.
Now, what's really interesting about this slide is seeing it in action. Look at how the parent amoeba stretches out, its nucleus divides, and it just cleanly separates into two daughter amoebae. Below that, you can clearly notice the horizontal split of the paramecium and the top-to-bottom split of that green euglena. It really is a beautifully precise mitotic division. But wait, what happens when things get tough? Like, say there is a severe lack of food, or the conditions are adverse. The amoeba actually stops moving and completely switches up its strategy from binary to multiple fission. Just focus on the top part of the diagram here. Let's walk through these steps very methodically. Step 1. The amoeba rounds up and builds this tough protective wall around itself, which we call a cyst. Step 2. Safely tucked inside this cyst, repeated nuclear and cytoplasmic divisions happen, creating many tiny amoebulae. And finally, Step 3. When favorable conditions return, the cyst simply breaks open, and all those tiny amoebulae are released out into the world at once. It's a truly brilliant survival trick.
Okay, let's transition now from those microscopic single-celled life forms to multicellular organisms.
Once we get to multicellular organisms, the methods get a bit more varied. Take the algae Spirogyra, for instance. When it has plenty of water and nutrients, its body literally just breaks up into fragments, and each single fragment grows into a brand new organism. Now compare that to regeneration in planaria, where if a body part gets cut, it regenerates the missing parts to form two whole, completely new animals. Or check out the budding process of the hydra. When conditions are good, a little outgrowth forms on its body, developing into a small bud. It grows its own little tentacles and eventually just pops off to live as a fully independent hydra.
Let's move to our fifth and final section today, looking at vegetative propagation and spores, things you might actually see right in your own homes or farms.
In the plant world, reproduction can happen using vegetative parts. We're talking roots, stems, leaves, and buds. We call this vegetative propagation. Think about the humble potato sitting in your kitchen. It reproduces using those little eyes on its tubers. Bryophyllum, a plant you might see around, uses buds right on its leaf margins. And farmers know that crops like sugarcane and grass propagate using the buds found on their nodes.
And this brilliantly illustrates exactly what we were just talking about. Notice those small depressions on the potato tuber? Those are the eyes, and that is exactly where new shoots are going to grow from. Similarly, look closely at the bryophyllum leaf. You can see small new plantlets sprouting right along the very edge, or the margin, of the leaf.
Finally, let's talk about fungi, like mucor. If you have ever accidentally left a piece of wet pakri or bread in a dark, humid place, you've definitely seen this fuzzy stuff. The fungus has a thread-like filamentous body with these little round structures at the top called sporangia. Once the spores are fully formed inside, the sporangia just burst open, releasing all those spores into the air. And whenever those spores land in a nice, warm, moist spot, well, they germinate, and a completely new fungal colony begins to grow.
We have seen today how incredibly fast and efficient asexual reproduction really is. It is absolutely perfect for multiplying rapidly. But I want to leave you with a thought to chew on. As we learned earlier, asexual reproduction gives us zero genetic mixing. So, what happens when the environment changes and a species literally has to adapt and evolve to survive? How does nature solve that massive problem? Well, my friends, that requires a completely different mechanism, and we will dive straight into that in our next lesson on sexual reproduction. Thank you so much for learning with me today, and keep observing the wonderful world around you.